This is the first lesson of Chapter 10. We're going to be continuing our discussion of how cells divide, making new cells. But unlike in mitosis, meiosis results in cells that are haploid. And these are these are cells that become the eggs and the sperm. And so let's take a look at meiosis. Similar to the mitotic cell cycle, meiosis involves several sub-stages. Also, like the mitotic cell cycle, there are interphase and then the phases of meiosis, as shown here. In fact, as you can see in meiosis 1 on the left, interphase is actually before meiosis, the first phase, if you could say. There is a first phase, because it's a cycle, of course. It kind of depends on where you're starting, what stage that cell's in. But there is one interphase throughout the entire meiotic cell cycle. Notice that. That's a very important detail. And then that is followed by meiosis 1, which includes prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. And then there's a second round of meiosis, but note that there's no interphase in meiosis 2. It goes right from telophase 1 and cytokinesis to prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. So now we're going to take a closer look at each of these phases. Like we did with mitosis, we're going to go over some of the key events and structures that would happen in that cell at that stage, and then make a drawing of a hypothetical cell. So when we look at the model cell drawn here, we're going to be modeling, like in mitosis, the animal cell with a diploid number of 4, or 2n equals 4. That means that a normal body cell of that animal has 4 chromosomes. However, the resulting cells from meiosis are going to have half the diploid number, and that is called the haploid number, and that will be 2. But we'll see that at the very end of the process. So remember that interphase involves 3 substages, G1, S, and G2, and they occur and have the same exact events as we learned about in mitosis. So if you want to review those, you will go back to the lesson on mitosis and see the details of G1, S, and G2. So the cell that I've shown here would have already gone through gap 1 and S. So this would be specifically in G2. And the reason I'm emphasizing that is because there's already been a duplication of the centrosomes with the pairs of centrioles in them. And also, although you won't know because we have no means of comparison by looking at this, but I want to also make it known that the DNA in the form of chromatin right now, the uncondensed DNA in the nucleus, has already duplicated. Now I'm showing 2 different colors here in the chromatin, and there's a reason for that you'll see as we continue on in the lesson. Okay, so in prophase 1, you see that the nuclear membrane is starting to disintegrate while the chromatin condenses into chromosomes. Now these chromosomes are in the form now of the duplicated chromosomes, or sister chromatids. But this looks a little bit different from mitosis because of the fact that there's 2 pairs of sister chromatids that are forming this association together. And that is called a tetrad. Those chromosomes within the tetrad are homologous. And what that means is that they code for the same genetic traits, like let's say hair color or height for a human. And the reason now I'm using the different colors, red and blue, is to represent the maternal and the paternal genetic material. Every animal has a mother and a father, and their cells have DNA from both parents. Each of those maternal and paternal chromosomes duplicated during S of interphase, and so this is why we have a pair of blue and a pair of red sister chromatids. And together, again, they form a structure called a tetrad. I'm going to zoom in and on the next slide to show what's happening in that tetrad and what this crossing over might be. But before I do that, let me finish talking about prophase 1, including the formation of the spindle as shown at the top left. So the pair of duplicated centrosomes, which with pairs of centrioles inside, now have started to migrate, and the spindle fibers are forming in between them. And the picture I drew would be more of like an early prophase, but this is going to continue to move. The spindle fibers are going to start to gather the chromosomes by attaching to their centromeres. So that's what's described in the last bullet point, even though that's not reflected in the drawing here. You might see early prophase, something like what I drew, or it could be a later stage of prophase. So now we're going to zoom into a tetrad and try to understand what that is and what this crossing over could be. Okay, so let's take a look at that tetrad. This would be the condensed chromatin forming a chromosome, but they've been duplicated and they're attached at their centromeres. And then, so those are the maternal duplicated pair of sister chromatids, and then we have the paternal pair. Each of these has a kinetochore at their centromere, just like again we saw in mitosis. These are special motor proteins that are going to be helping those sister chromatids separate and then also become daughter chromosomes ultimately in the final daughter cells. So again, these are centromeres, which are the central part. That didn't come out too great. Let's see here. Okay, that's a little bit better. So now you can see I've labeled the centromere, which is the central region where the pairs of sister chromatids are attached. And at each centromere is a kinetochore protein. And I've also added a note up at the top that this whole thing, this association of two pairs of sister chromatids, is called a tetrad of homologous chromosomes. Okay, so the next thing I want to show you here is something that could happen. Sometimes where you have the overlap of the maternal and the paternal chromosomes, there could be this phenomenon called crossing over, where basically they swap places. A little bit of the blue will switch places with the red piece. And this is a very important phenomenon that can actually lead to an increase in genetic diversity of the daughter cells. So I'm going to be showing this crossing over for the rest of the steps. So I'm just going to show here that it happened. Imagine that they've already switched places. And so in the remaining steps of the process, we're going to show that that crossing over has already taken place. Okay, so I'm just going to make a note here that crossing over leads to an increase of genetic diversity of the daughter cells. Also, crossing over can only take place in prophase 1 of meiosis. So you're not going to see that happen again, even though we'll see the results of that carry through to the later stages. All right, so here we have metaphase 1. Notice that the tetrads still together have lined up on the metaphase plate, which is that imaginary equator of the cell, and it runs perpendicular to the direction of the spindle fibers coming from the centrosomes. So at this point, the spindle microtubules have attached to the centromeres of the sister chromatids and lined them up. I also want to point out that on the long set of chromosomes on the left, I've tried to show crossing over there. So that already happened, remember, in prophase 1, but I'm just going to continue to carry that through till the end of the process. Moving along, we have anaphase 1, where the homologous pairs within the tetrad have separated now into the sister chromatids. So we have, for example, on the left, the long pair of red sister chromatids, where we do have a little bit of blue from the crossing over, has separated from the blue pair of sister chromatids with a little bit of red. And then on the right side, we have a shorter pair of blue sister chromatids that separated from the shorter pair of red sister chromatids. And they're being pulled to opposite sides of the cell as the kinetochore microtubules shorten. Those are the ones that are attached to those kinetochore proteins. Remember, there's actually, I'm not going to show them for everything, but there's proteins there that these spindle fibers of the spindle are attached to. While that's happening, the non-kinetochore microtubules lengthen, and this is what's causing the cell to stretch. Notice how it's looking more like an egg shape than perfectly round. In telophase 1, you will no longer see the spindle fibers because the spindle has disintegrated, as both of the pairs of sister chromatids have fully made it to the opposite poles of the cell. And while that's happening, cytokinesis occurred, where the cytoplasm divided. And that resulted in 2 independent daughter cells that we have here. Each daughter cell right now has 2 pairs of sister chromatids, a long pair and a short pair. And see how the long pair has that little bit of crossing over that took place in prophase 1. So we did see it follow through all the way to the end. So we're going to take a pause here for this video because we've just completed all the stages of meiosis 1, and we'll pick up in our next lesson with meiosis 2. So recall where we left off and be ready to go through a second round, which ultimately ends with the daughter cells that are going to be haploid and will become the eggs or sperm for the animal that they're in.